go first?
Sure.
Yes. Okay. Do you want a background of what the grant's about, or you just want to know what the experience is about?
Well, you can tell us a little bit about your project and then
Okay.
about, the experience.
Yeah. Okay. So my project is basically we're trying to make these mini brains. The reason we're trying to make these mini brains is because there's a lack of knowledge on how the immune system responds in the brain. Right. Up till now, there's been like that's a no-no. You don't want the immune system in the brain. We've done tons of studies in the mice. We get both good stuff and bad stuff, but no one's been able to do it in humans. So I had this idea, let's make a human brain out of an individual and get his immune cells and plop them together and see what happens. A little more than just plopping together, but y'all get the gist. Basically, I applied to the ESI last year. I got it. The experience has been great. The money came in when it was supposed to. The only issue I think we had, and it was like hiccup, is confirming the whatever we purchased got applied to the right account. But because we use a database here in my lab, we were able to coordinate and show that this is what we bought, this is what is going on. Right now, we're halfway through the project. We have ISPSCs. So one of the things we want to do is get blood from human subjects, drive them to ISPSCs, and then drive them to these mini brains. We're at the stage of the ISPSCs. Hopefully in 1 or 2 months, we'll start having the cerebral organoids. The goal of that is to get those pictures and show the NIH that I can do this because this is really novel technology. And actually, up until the deadline point, whatever pictures data I have, I'll be putting into my 2 grants, my KO1 that I'm submitting in November, or resubmitting in November with Paula's help, and then an RO1, and I believe that's in February, if I'm not mistaken. But yeah, we're using all the data we're getting out of the ESI grant to generate preliminary data for the grants that we're applying right now. Yeah. I can't hear you. I can't hear you. Y'all
There I am. There I am.
There we go.
So
Okay.
just to clarify for everybody, the early stage investigator awards are for $75,000 for 1 year. The next application is November 15th. And we go by the guidelines of NIH, what they call an early investigator. So within 10 years of your PhD. So we're hoping to kind of drum up some more business. We have one successful applicant, and we were very lucky because just as Sterling kind of was coming online and Dr. Griner with their grants, Meredith started running the program. So because she has that MBA, she's kind of smart about managing money and helping make sure you guys get your tranches of money, which was totally baffling to me. So Stacy, you want to tell us a little bit about yourself and your project and your experience?
Absolutely, I'd love to. I made something brief just to keep me on track. I was not fully aware if I was coming today because I was on backup for IPE. So I was counting down the minutes till I found out and keeping my fingers crossed that I wouldn't get selected, and I didn't. So I'm super excited to be here. I am Stacy Griner. I'm in the School of Public Health. I do research on sexually transmitted infections, oral health, because I'm a dental hygienist in a past life, and implementation science, which is a form of translational research. So my project is about the determinants of implementation of prenatal syphilis screening guidelines. So we have both guidelines that come from professional organizations and policies that come from a Texas state level on what providers should be doing about syphilis. In the past couple years, syphilis has increased about 650% in Texas. It's wild. It is out of control, and it's especially prominent in pregnant people, which leads to congenital syphilis and has some really negative impacts on moms and babies. So I applied for the early stage investigator mechanism because I had 2 RO3s that were reviewed and scored about STI screening during pregnancy broadly. But in talking to a program officer, like, help me get this funded, they said it's difficult when you're looking at STIs broadly because you can speculate about the impact. We don't know what direction you're taking. It's too broad. And they recommended targeting syphilis. But I have no expertise in syphilis. I've not done any work on syphilis. Most of my work is on chlamydia and gonorrhea. So I thought, let me just give this a shot and see if it'll help. I have been using implementation science approaches in this project to understand the barriers and facilitators to translating these guidelines and policies into practice. we've so far conducted 10 interviews with prenatal providers. Most of them were certified nurse midwives, and most of these were conducted in April and May. So because I got started and was able to do some interviews early, I put in an RO3 in June using this as sort of preliminary data. Again, it's still an RO3, but it will be reviewed next week. So everyone just keep your fingers crossed. I feel really good about this one, really, really good. But I've been wrong so far, but you never know, things might change. It'll be reviewed on the 25th and the 26th, I think. And some of the co-investigators on this project are Dr. Allison from the Center for Health Policy and Megan Nelson from TCOM, who is a prenatal provider. I am also disseminating this work at the annual Dissemination and Implementation Conference. It's in December in DC. I am very excited to go there. I've been for the past couple of years, but I'm excited to take this work with me because they have a very strong maternal and child health section. So I'm excited to make some connections and get some feedback on the project and where we're headed, to find out if I'm missing anything and just have a conversation around what's been done. So again, my co-authors are two doctoral students from School of Public Health, two DO students, and Dr. Allison from the Center for Health Policy. My next steps. So we've completed everything we said we were going to do, but I am going to do just a little bit more. So
Yay!
yeah, I have an opportunity here and I don't want to pass it up. So I initially went in to recruit MDs and DOs and also certified nurse midwives. It's been really difficult getting physicians to participate in the interviews. So I've expanded the recruitment. I just put in the IRB amendment last week to focus on more mid-level providers, so NPs and PAs. So I think that'll give us more of a comparison to the midwives, and we'll have a better sample that we can report on. So I'm excited about that coming through. I'm still waiting on the IRB. I want to do about 9 to 10 more interviews by the end of the year. I think that's feasible given the trajectory we've been on and how well recruitment's gone. My plan is to spend January and February finishing the analysis and continuing to write up the manuscript. So we're doing pretty good. I think there's an opportunity to expand it a little bit more, but just want to quickly to say thank you to everyone who took a chance on me in this project. I do think it's been really helpful in my career, and hopefully we'll all have something to report on about it. And pretty soon you'll be saying, wow, look at this early stage investigator who got two early stage investigators who got RO1s off of this project. So I hope that I can bring some success from this project too.
Stacy, thanks. So now I want to open it up if anybody's got any questions. I know there might be some people on here who are thinking about applying for this mechanism, or not. And Stacy, one thing that came to my mind was, if you have demographics, is there any health disparities that you see? Because that could be a whole nother avenue of funding.
Yeah, that's true. Most of the people who I've talked to, because I'm focusing on providers, they're mostly white women, especially in like maternal and child health programs. But I think you're right. I think there's a lot of opportunity, especially with syphilis, to do health disparities. I have lots and lots of ideas, but I got to do one thing at a time.
Awesome. Excellent. All right.
Okay.
Well, thank you guys. Yeah,
Okay,
so
I will share my screen.
we're going to have our next presentation, Dr. Dongming Su, who applied for the bridge funding. Wait.
Can you see my screen?
Yep, we can see it.
Okay, that's good. So in my okay, I can see it. Yeah. Okay. Anyway, so anyway, my name is Dongming Su, and I'm in the Department of Microbiology and Immunology. So I luckily got the bridge funding support through this year. So I also was told about the not to introduce this research, just introduce about the background and the plan. So I think that this bridge funding has really supported my research bridge. So first, I want to greatly thank the research enhancement assistant program committee. So the background, so I have last 5 year NIH support and 1 year non extension, total 6 years, and this funding was almost about to run out at end of last year. So at that time, I'm very nervous. I think I still had a student, PhD graduate student, and still less 6 months for graduation. So, and the paper, his paper was not published at that time. So very luckily, we got just before my NIH funding run out. So in October 2021 last year. So the REP announced the bridge funding program. So I immediately submitted my application, and I was lucky enough to obtain the award in the second month. That's January this year, January 2022. So this month, this funding actually that's really saved my research project. So during within 1 year bridge funding support, so I make my student, PhD student, graduate successfully within 6 months. So I paid his tuition, his stipend, and published his paper. So we published the paper in a high impact factor journal. That's in the agency 11 impact factor. That's 11. So in this paper, we acknowledge the UNTHC bridge funding. So that's the funding support me, and then I also acknowledge the funding. So during this year, I also submitted 2 NIH R1 grant. That's based on this funding. We made sufficient preliminary data. We submitted 2 NIH R1 grant application. One is submitted on May. Another one is on the this June. So in addition, we still have several months to finish this grant, this project. So we still make an effort to generating the preliminary data for another NIH grant. That's your one, that's specific for the RFA. So I try to submit as deadline is next year February. This is general background of the plan. So for we also get the emails that talk about our experience how to get this application, get this funding, get this application done. So I cannot say my experience that's suitable for everybody. So, but at least for my situation, I think that you should write the SMART goals. You have to write it SMART. So SMART goal that means specific and measurable and achievable and realistic and time bound. So based on my experience, so specific, that's 1 page, this SMART. Specific, you don't need to write the specific aim in this item. So because you have a specific aim page, you write clearly what you want to do, what you wanted to reach. So, and in this part, I think you just say your specific situation. For example, my situation that's I shortage of funding, so I shortage of sufficient data for submitting this funding. So NIH funding. So I need completing this insufficient experiment, and then to award any discontinued for my ongoing research. For example, I explain the animal. So animal model, if I have no money, the animal will terminate. And then if you want to purchase a new animal or set up a new model, that takes time. So maybe affect your submit your NIH grant, and then also personal. So I just mentioned that I have a PhD student, just shortage 6 months. I need this funding. So that's specific for your specific situation. So second one for the measurable. So you should mention, for example, your result data. That's from your design experiment. That's measurable. You should also, if you have publication with this grant, or you have, of course, the purpose for the submission of the NIH grant. So that's all the measurable. So you should mention. And the charitable, I think you should mention your previous experience, publications, and any unique research model, something that's this ensure your goal are achievable. So for the realistic, so I think also you should mention your experience, and also you should mention about the research project, including ideas, models, and approach and reagent. That's all within your area of expertise. So in other case, so you also should provide the core facility to support you, and then provide you any advanced technical support, and then this experiment or your design experiment or design aims are realistic or feasible. So for the time bound, you just, for example, I mentioned 3 months, I want to finish something, 6 months, half a year I should finish something, and then final should do something. And this is similar to your NIH grant. So that's called a timeline or one, but the NIH grant one is 5 year, 5 year milestones. This is just 1 year what you will do. So that's my experience, not correct. So in brief, I just want to catch up with something. One is the gap grant. So the bridge grant, bridge grant, you just need you must have the new gap, 1 year and then 2 years gap. So you cannot bring your 4 years ago, something 4 years ago, bring that's maybe not supported. I mean, the LEAP will not support you, I think. And then another one is ongoing experiments, not design any very any a new experiment. Of course, you want to find a new issue, new evidence or new data, but this is still connected with your previous experiments. So second, the third one, I preferably you should have the current personal. So for example, I have a PhD student in my lab, or technician, or someone, and just need to continue. If you write the gap grant just 1 year, you said, oh, I wanted to recruit a new postdoc, that's maybe they will not give you fund. If the put the postdoc for the new recruitment, so you get the people, get the person coming in, and you need training or grant already gone. That's 1 year already done. So if you have the scored NIH application, probably recently application got scored but unfundable, and that's very good. Or get the comments, the critics write something, said your application not have sufficient data. That's a very good reason. Is that I need to generate establish sufficient data, and then I need money to finish this this NIH grant. So get a successful funded. So that's only ask me to talk about 5 minutes. I think 5 minutes already done. So anyway, the final, I still need many thanks to the research enhancement assistant program committee. Another issue, I wish everybody good luck to get your application done. Okay. Any questions?
Well,
Yeah. Any questions?
thank you. Thanks, Dongming.
Okay.
I'm going to say that Dongming was actually kind of our real guinea pig. We rolled out the bridge grant mechanism as the first mechanism. And for those of you that don't know, it is, like he said, it's to bridge and keep things going when you're between grants so that your students can still get paid. You can keep your animals or whatever. And it is on a rolling application basis. So it is not, just once or twice a year like the others, because again, you're never 100% sure when you're going to need it. The other thing that we do require in this is that you get some help in, writing your resubmission and writing your introduction. I'm going to tell you, Dr. Tom Cunningham is like, he has like a wizard hat. I've never seen anyone could do such a good job with helping people write their introductions successfully to get there.
Yeah, that's also I should mention. That's Dr. Cunningham, and you also helped me a lot. So write your draft, send it to them to let them to give your comments back and revise. That's very helpful.
Well, and I heard that from your chairman that you have just gotten every penny out of this grant, which is exactly what we want to happen. So thank you again. Thanks for being the guinea pig. And thank you for being, look how much you have accomplished so far. Dr. Ma, what you got to say?
Yes, I'm probably is another example. Let me get my screen shared.
All right.
All right. Okay.
Wow.
I have 10 slides.
That's more than 5 minutes.
Well, thank you very much for having me here. As Dr. Su mentioned that, well, I'm not sure that my experience apply to everyone or not, but I do love to share my personal experience with everyone in applying for this grant, and also some message during the process of this application. Well, as everyone knows that this program is designed for helping faculty members who may have the budgetary gap in research, which actually most faculty members may have in certain period of time. Typically for those faculty members who recently submit an extramural grant, but it was scored, but did not good enough, but was not good enough to get it funded. So the BFP, the mechanism can help you to generate the more preliminary data to respond to the critiques and help you prepare for resubmission. In my case, I did submit an R1 last year, probably February, and got a review back in sometime June. And it was scored, but exactly, and as mentioned in the program announcement, is not good enough to get it funded. As you can see that 36 percentile. So I thought that I may fit the criteria of the BFP program. And so I decided to put one in. And the next thing is that, well, what I'm going to do with the funding if I get it. Well, as mentioned by the program, is that way to help the faculty members to generate more preliminary data and to prepare for resubmission. So I looked over my summary statement. Well, of course, what you can imagine that there are many critiques, but in my judgment, and the one major critique from the review number 1, my original hypothesis in my original proposal is that a small protein called IMFA, which can stimulate renal inflammatory response by stimulating the production of cytokine and chemokines by kidney cells. Of course, I presented some preliminary data. In my preliminary study, and I used ELISA kits, which can simultaneously measure 36 cytokines and chemokines, and in response to kidney cells, which manipulate the IMFA expression level. And then in my proposed study, I selected the responsive cytokine and chemokines and found it in my preliminary study. And I'm going to focus on this responsive cytokines and chemokines in this proposal. One reviewer mentioned that he liked this idea, but he mentioned that the ELISA kits, 36 cytokine chemokines, may be too narrow. So he or she suggested that we're using the RNA sequence approach to screen the cytokines and chemokines in response to IMFA expression. Of course, this is a good idea, but you can imagine that it is a very expensive study. So I thought that maybe I can use the BFP, the funding, to take care of these critiques. So I put the M1, the specific M1 in my BFP application, and I propose that I will do the single cell RNA sequencing of the isolated kidney cells from IMFA wild-type mice and IMFA knockout mice, and compare the gene profiles of the cytokine and the chemokines between the wild-type and the knockout mice. So this is the M1. We made some progress in this M, and we did collect the samples of the isolated single cells from wild-type mice and IMFA knockout mice, and we have this model. And we submitted the samples to the genetic core facility in June. And our genetic core facility collaborated with 10x Genomics do the single cell RNA sequencing. And this was completed just last month in September. Right now, they are working on the data analysis, and they work very hard. It's kind of a time consuming, the huge data. So I hope that we can get something out very soon. Another major critique from my original proposal is that one reviewer was not very happy with my animal study. I proposed a signaling pathway in my original proposal. I proposed that IMFA stimulate renal inflammatory response through the beta-catenin and the alginine pathway. One reviewer felt that what this animal study did not develop very well. Basically, they wanted to have a more preliminary study to support my hypothesis, this pathway. So I thought that, well, I can put this as a specific M2 in my BFP application. So what I'm going to do in the BFP application proposal is that I can collect kidney samples from the animals treated with the nanoparticle IMFA siRNA. We have already established an in vivo delivery system which can carry nanoparticle siRNA complex specifically delivered to the kidney cells. So we can use this approach and deliver nanoparticle and IMFA siRNA to the mouse kidney and kidney cells. And then we can collect the samples and analyze the protein expression level of downstream proteins. We also made some progress in this specific M. So now we have already collected kidney samples from the from the mice with and without the nanoparticle siRNA treatment. And the next step would to do the immunohistochemistry examination and to analyze the downstream the protein expression level, such as beta-catenin, the alginine, with and in the kidneys with and without the treatment. So this BFP program really helped me to generate the preliminary data and to help me prepare for resubmission. Another information I would like to share with everyone is that this program not only help us financially, but also help us scientifically. What I mean is that when you apply for this grant, as required by the program, you have to submit the previous criteria with your proposal. In the proposal, you have to describe how do you respond to the critiques. So our reviewers, I say our reviewers and our colleagues will review the critiques, your previous granted critiques, and your response and how to address these critiques. So they will give you comments. They will give you feedbacks that will help you to prepare for resubmission. It was very true in my case. I got very helpful tips from our colleagues. For example, I just quote some of the critiques, our reviewers. For example, one reviewer mentioned that, well, Dr. Ma got 36 percentile score, didn't get what fundable level. The major reason he mentioned that probably from review number 1. So that reminded me that in my resubmission, and I probably focus on review number 1 to deal with this review and very carefully. Another comments from our colleagues that is about my productivity. This is not concern from my NIH reviewer, but it is kind of a red flag for me, and it may be a concern for next review, for next cycle. So that reminded me that I needed to fix the hole before I resubmit it. So which I did, and I can use this period of time to my resubmission and increase my productivity so I can fix the hole. Another example is that one reviewer mentioned that, well, so give me some suggestion. Well, said, well, beta-catenin, because I proposed the IMFA, beta-catenin, and then the alginine pathway. And one reviewer mentioned that beta-catenin has so many functions involved in so many pathways. So that is something we may need to take into account. So that reminded me that when I do resubmission, I probably pay more attention on this pathway and maybe discuss more. So in summary, when you decide to submit the PFP, the application, and first of all, what we do step by step. First of all, consider the eligibility, whether to fit, and then what are you going to do with the PFP the application, and how to do it. You need to describe and how to respond to the critiques. And lastly, no matter you get it funded, and take our colleagues' feedback positively, because they really want to help you. And finally, I would like to take this opportunity to thank our VPR office and the support our research is very helpful, and also our department, because we're the one third of the funding from the department. I really appreciate our department and our chair, Dr. Kuhn, right here. And lastly, also appreciate our internal reviewers. They give me very good comments and feedbacks. Thank you.
Thank you. That was fantastic. Okay, so if you're not intimidated already by their presentations, you may have some questions for Dr. Su and Dr. Ma. I think they did just a fantastic job, and we're going to pay you some money later. No, I'm just kidding. Oh, they're so good. It's so fantastic. But that was really helpful. And just to, see where you're going with this, how much you've accomplished, and that this is a really important bridge to keep your research going. Any other questions? Anybody? No? All right. Well, thank you again. And now we have the winner of the Team Science Award, the PI of the team. And so this is Dr. Jonathan Toon. The Team Science is a bigger award because the idea is that we're trying to foster collaborations that could end up with program project grants, multi-PI grants, those kinds of things. And so in our first go round, I think we had 5 or 6 applications for the Team Science. And the winning team was Dr. Toon's team. And so he's going to tell you about his experience.
All right, thanks, Paula. You can hear me okay?
Yep.
All right. So just kind of take a little bit of time to tell you about the sort of forming what we've called the Translational Cardiovascular Research Consortium. So I'll sort of set this up and how this kind of came about. So with the opportunity and coming in the door of really wanting to set up a more team-based science kind of thing, and with this opportunity from the school, sort of sat down and thinking with others in collaboration first about question, right? What's a significant question in the field? What are the barriers, potential solutions to that? And how can we leverage existing strengths within the department, the school, the institution to begin to address that question? And then starting to reach out to individual investigators, key stakeholders, other people in the community to see, can we build this team around the question that's working from our strengths, and then again, ultimately building this team around this thing. So this was kind of the kind of iterative framework that went into establishing the team and beginning this effort. And really just like real quick, I don't want to get too deep in the weeds about the science, but the question that came up for us was the issue of heart failure. It is an existing and growing problem. 6.5 million Americans have heart failure. The number is growing every year. There's increasing recognition of different types of heart failure, and one that's called heart failure with preserved ejection fraction, or HFpEF. It's now about 50-50. You know, classically you hear about reduced ejection fraction and big dilated reduced ejection fraction heart failure. It's now about 50-50. And it's on track to becoming the most common type of heart failure moving forward. And the cost of this to our economy, to our society is huge and growing. And more importantly, I think, are the outcomes that are tied with heart failure. So here is just a little bit of epidemiology data on, again, about this 50-50 and a few kind of fitting in the middle. They kind of go either way. But the biggest issue with this is that the 5-year survival of heart failure patients, in particular these preserved ejection fraction patients, after they're hospitalized, their mortality is worse than almost every type of cancer out there. So, and the other thing is, so this has been increasingly recognized in the cardiovascular field, and that's led to a lot of discussions in the literature. So here's a 2020 paper from Circulation about what are the research priorities around this question? What do we need to do and what's going on? So the big take homes of this big working group, so these were top cardiologists and scientists from around the world. The take homes from this working group paper were, first, that this is a major problem and it's rising in prevalence and it has high morbidity and mortality. And furthermore, and I think even more importantly, there are no known effective treatments for HFpEF. So everything that works clinically, medicine-wise, to treat patients to make them better doesn't work in this group of patients. And so because of that, and here's the big question, HFpEF and related questions is now considered to be the greatest unmet need in cardiovascular medicine. So these are take-homes from this working group. So, and in that paper, they identified key barriers and potential solutions to addressing these huge unknown questions. And the first thing that caught my eye in trying to build on that was inadequate preclinical models, because our group has been working on, for the past few years, a swine model of HFpEF. And so leveraging that existing large animal model with, again, other things here at the Health Science Center and local hospitals to begin to say, can we build a cohesive clinical and basic translational team to begin to address these questions? So this is what really formed the framework. around building the team. And again, that was kind of built off of this recent paper that we published in our Ossiball swine model. So really fast, we have a unique swine model. These are Ossiball pigs. Interesting story, but what we found in these pigs, in either lean or if we feed them a high fat, high calorie diet for a while, we overpace them. We pace them at 180 beats a minute for 4 weeks. They get full blown HFpEF. They get sort of their right heart, their pressures go up. There's evidence that their capillaries drop out, that they lay down fibrotic, their hearts become more fibrotic, and their heart function here, this is compliance. So this is pressure relative to volume. They have elevated pressures regardless of filling volume. So this is classic heart failure with elevated pressures, and their ejection fractions are perfectly normal. So this fits this sort of clinical definition of heart failure. So we have these studies that are ongoing. We have tissues from these animals and lots of questions. So for example, we're talking to Dr. Ma now about looking at the kidneys from these pigs, right? Again, leveraging strengths of this and trying to build team moving forward. So, but with this pig model, we initiated conversations with local cardiologists at Medical City, Fort Worth, who are interested in having their residents participate in studies here at the Health Science Center, who are experts and then can identify human subjects. So working with the Romero and Ricard's lab, and also with Mike Smith. So we've, and I'll show you in the aims in just a second. So initiating human subjects, like pilot studies to assess cardiac and autonomic function in heart failure patients. We can do similar and have done similar studies in our pigs, so we can then compare. The other thing that we can leverage here at the Health Science Center is that the Willed Body program here in the anatomy department in the Center of Anatomical Sciences has really exponentially grown over the past few years. They're getting close to 1,400 to 1,500 bodies a year. Many of those have heart disease, still the #1 killer of Americans. So we are able to get tissues and begin to establish a biorepository of tissues, heart, brain, skeletal muscle, kidney, all the things that we can begin to do histological analyses on, to ask a variety of questions regarding capillary density, fibrotic disease, underlying histologic changes that occur over the disease state. And also with samples that we get from our swine studies as well as humans, begin to do some omic studies, kind of like Dr. Ma was talking about, to identify novel pathways that may be involved in the disease, because there are so many unknowns here. So that's how this sort of team came together. So with local cardiologists, with our large animal model, human studies, biorepository, building this up to ask questions in a vertical manner. And so the goal of this at the end of the day is to make advancements and improve understanding around this disease of HFpEF, if you will. So the major aims really are to begin to collect data, expand. So we have the pig studies going, but expanding this to humans, like how does this translate out to humans? So aim 1 is to begin, and we have, again, we're currently writing the IRB protocols to start assessing cardiovascular and autonomic function in subjects, human subjects with heart failure, and these will be identified by the cardiologists at Medical City. They're going to send fellows over to help with these studies. So that's the first part. The second part is we have banked back tissues from our previous swine study with the failure. So we can begin to assess cellular molecular pathways, do omic studies that may be contributing to the phenotype that we're seeing in these pigs. And then the third part is to develop that biorepository. We have the pigs, we have the human samples that we can get from the Willed Body program to begin to ask these more detailed questions about this. So again, sort of vertically up and down, large animal model, human studies, biorepository, to address those barriers / solutions that were laid out in that white paper. So with that, how this all kind of comes together is that there are key phenotypic features of heart failure around changes in heart rate and how it responds to pressure, exercise, etc., cardiac function. There are underlying comorbidities that are seen with this disease. There are new treatments. These are SGLT2 inhibitors. This is a drug that's used in diabetic patients that's been shown to mitigate progression of heart failure. And we have preliminary data in our pigs that it can really slow this down. So we want to use this agent to see if it can slow this progression down. And then with the more cellular molecular kind of things, begin to ask mechanistic questions of what's going on in the heart muscle itself with calcium handling, changes in proteins that affect cardiac contraction, and again, sort of across the board, up to humans, pigs, and then down even to cell molecular type pathways. So that kind of in a nutshell is how this came together to leverage this, what we hope really becomes where we're coming in with multi-PI grants and ultimately program project type grants where we have human studies, large animal studies, cell molecular kind of things to really build this out. So hopefully that's clear. That's
Awesome. Thanks, Jonathan. So I've got a question.
shoot.
I heard that that school over in Dallas, that they have some expertise in cardiovascular. Is that a concern, an issue? Or...
Actually not a concern, actually something that we're planning to leverage. So Stephen Romero, who's a key part of the human studies side, he's actually a co-investigator with that group that has a heart failure PPG. The questions they're asking with their group are very different than what we're asking. And they're actually willing to help us identify patients for the trials that we're doing here. So I think the ultimate goal is to work with them, right? And what they're doing so we can work together, not against each other, right?
Fantastic.
So good question.
Awesome. Okay, and I have one more question. So where I used to work, they did some cardiac fibrosis studies with alcohol because it's New Orleans. Have you guys thought of throwing some alcohol in here or looking at alcohol in your patients?
Haven't done that. I mean, I think the big thing that we've done is obesity, because a lot of these patients that get this HFpEF type phenotype, it's at least there's a lot of question around if the obesity pandemic is driving a lot of this increased prevalence of this disease. So we've been a lot focused on that side of the question, but yeah, haven't done alcohol, but people like that too.
Yeah. And IAAA is pretty good to get money from. So yeah, just saying. Other questions? So the team science grant proposals are once a year. Meredith, do you know when the next deadline is going to be, or do we even know?
We don't know yet.
Okay,
We're waiting
good.
for final approval.
I was like, I don't think I know this.
No, we don't know that yet.
Gotcha. So it's something to think about. Obviously, we have some strengths and some themes here at the school that people can take advantage of to put their team together. I really thought it was very exciting to have this beginning of translational science here. I'm going to call it a beginning. I know you guys are more than that, but
no, I think that this is a great opportunity to seed this type of research. And I think, so we're very appreciative, one, of the opportunity, and two, I think very optimistic of scholarship to come out of this, right? As well as the opportunity to apply for funding moving forward. So yeah, it's great.
Awesome. Yep. And I think, yeah, so I was the SRO on that grant review. And that is one of the things I want to really stress. We're working really hard to make sure that the review is as transparent and as, equitable, et cetera, as possible. Because I think there's some history where maybe it wasn't quite as.
Yeah, I think, as Dr. Moss said, I'll sort of echo what he said. And so we got detailed critiques from, I think, 8 or 9 reviewers that were on the panel, and I think very helpful. And it highlighted some things like that we knew that this was a kind of an issue going in, and it just kind of highlighted, yes, this is things that we need to think about moving forward and make sure we're addressing those as, the studies progress. So I think very helpful looking at this from a constructive, positive lens and not the other way around. Right?
Yeah. And we're working really hard to add outside reviewers, because especially for team science, if everybody's on the team, who's left to review?
It was a challenge, yes.
So if you have ideas of people who have expertise in a certain area that you might be willing to review, because you're not going to have to review, but maybe 1 grant, maybe 2. Please let us know. Let Meredith know. I sent her some LSU people that I think could help. So again, we're really working to make the review as positive and constructive, but also as fair as possible. Because there were some real good applications, I have to say that. No other questions? Am I the only one that can ask questions? All right. So again, I'm just going to say the ESI.
Paula, I'll
Yeah.
ask a question.
Yeah.
I'll ask a question. So with the team science, and I got back 3 very detailed reviews on ours, and the rest of them were just scores. One question that I have with regard to this is that the team science grants have been set up to be very ambitious with respect to the goal being PO1, UO1 type grants. And there's something between an RO1 and a PO1, which is collaborative, multi-PI grants, that sort of thing.
Yeah.
And where is the space for that within the REAP program?
I think that multi-PI fits within the team science. The team science is kind of that preliminary step. No one is expecting Dr. Toon to have a program project as his.
Well, that was one of the comments I got back on our team science one was something to that effect. So, I mean, I don't know if they didn't like the team that was picked or the particular project, but the comment was that this is not something that, looks like it could be because of the people, because of the project, that could be going towards a large center type grant. So just something to keep in mind with the reviewers moving forward. In particular, because of the size of there was 1 award and it was a fairly substantial award.
Yes.
And going forward, I think you'll probably talk about the other kind of grants that there might be for the mid-career people or for that sort of thing, because those are going to be difficult situations as well, that if you don't have a score, you don't have a bridge grant possibility, you're not an early stage, so those are things as well to keep in mind.
Thanks, Derek. That just introduced what I wanted to talk about. So today at the Research Advisory Committee, we're going to hopefully vote on changing the ESI grants for the next go round to new investigator. New investigator just means that you have not had any substantial federal funding. So no RO1s or major NIH grants. And that will kind of fit more people. It doesn't have the time limit, et cetera, that the ESI grant, but the ESIs can apply for this as well. I've been trying to target junior people and say, hey, if you're interested, did you know about this? Can I help you write it? To do whatever I can to help them get some money and write a good application and get going on this. So I did, we've worked on that. Hopefully the RAC will approve it, and that will be for the April 15th deadline. Okay, well, look at that. 1 hour, we finished. Yay. Thanks, everybody. And thanks to our presenters. Thanks to our winners.
You guys.
Thanks to everybody.
Thank you.
Be sure to apply often. Whatever. What do they say?
Bye, y'all.
Thanks.
Thanks for the education, everyone.
Thanks.